This is a package on the right here. It's basically a wooden box with screens on both sides. 3 pounds of bees, newly mated queen, which you can't see here. The bees are all covering it. She's in a California mini queen cage on a sort of a tether that's right here. The tether is attached to the top of the box. And then this can here, which is filled with syrup. So those are made up by the producers by shaking bees out of many, many hives. These hives may have been involved in pollination of almonds and things like that, but they have it on a scale and they'll shake bees through a funnel and then add a mated queen and a can, and then attach them together for shipping either by pickup from the supplier or else you can go get them. And that's what we're doing as a club. There's a couple ways of putting the packages in your hive or installing. There's the shake method, where you literally turn the box over after getting it all set up and you dump the bees in the box. And the second way of doing it is called the pheromone method, where you set it up like this with your package inside and your queen between frames, and then you close it up and the bees migrate out of the box to be around the queen. So as part of the initial management, you want to delay the queen release for about 2 to 5 days. And we do that with candy in the queen cage. The California queen cage, mini queen cage, has a hole in one end and a cork. And when you get it, you take the cork out. And I want you to look, if you're new, there's going to be some videos I'm going to give you. And the Be Kind installing a package has a segment where the guy takes the queen cage and he shows you, look at the queen, where is she when she's on the other side? You pull out the cork, put your finger over the hole, and then when the queen is on the opposite side of the hole, you take your candy and you put it in the hole. He uses a plastic tube that has candy in it. We're going to be using gummy bears. We'll hand those out when the packages come. So the reason for delaying the release is the queen and the bees don't know each other. They're not sisters, they're not related. They just get dumped together and they need some time so that they don't kill the queen. That takes about 2 to 5 days. Once you've got your package going, you need to feed sugar syrup. If you're new, have new hive, you don't have any drawn comb, you kind of need to give them enough carbohydrate in order for them to build comb. It takes about they say a 1 pound of wax takes about 8 pounds of sugar. So anyway, you're going to learn all of that. This is just quick here. There's many ways to feed. I have a slide on that. And then you have to look at proper frame spacing. You don't want the frames pushed apart. They have to be tight together, because the Langstroth hive, if you are using a Langstroth, is designed specifically to give the proper bee space when the frames are pushed together. And then finally, whoops, where am I? Finally, you want to make sure you don't have Varroa. You hope and expect that the bees that are coming from the providers are relatively clean and disease-free, but these are bees that are have been around for a while. They may be new. They may be new nurse bees. They may be older bees, and you don't know if they're actually free of Varroa. Most likely not. So at some point, it's good to figure that out. So I talked about feeding. These are methods of feeding. here is a funnel going into an internal feeder. Elizabeth loves this method. You just fill it up, the hold whatever the size you buy, and you have to remove a frame or maybe two to get the feeder in, and then there's ladders here that the bees can climb on so they don't drown. This is a Boardman feeder, goes in the front. Many people don't like these. You know, it's the old, you know, ask 5 beekeepers, you get 10 answers. But there is a sort of a consensus that these can lead to robbing because the sugar is accessible. This, I'm getting feedback. If everybody could turn off your, mute yourself. This is just a jar that's in a hole that's drilled in this board. The board just goes on top of the box. And then finally, this is a box with the package in it, and then jars with holes in the lids, and then another box around it, and then you'd cover that up, and that's another way of feeding. And here's another way. This is, you know, solid sugar, driver sugar can be used. And anyway, the bottom line is your bees need to be fed when they're coming in a package. They don't have enough for themselves. So what are your objectives? First of all, rapid comb construction needs feeding. Second, egg laying and brood production. And finally, you want low Varroa numbers. You're probably gonna have questions, right? What's going on here? So if you need information, you can find detailed information about the packages. This is the page for the website, for the SMBG website. I've clicked on store, and this is the page that comes up. If you go down to this, there's 3 boxes that are blue. The middle one is package B information. If you click on that, there's a little message there with a link to the frequently asked questions. You click on frequently asked questions, and you'll download it's about a 3 or 4 page information sheet. It tells you when the bees are coming, how to come and get them, where to come and get them. All of these things are detailed on the frequently asked questions sheet. And then finally, there is, I guess I got to go back to this one. So that's one way to get the information from the Bee Guild store. Another one was Alex put it in the February 2022 newsletter at the bottom of page 3, all about packages. Pardon me. Wayne, I think you're not muted. And there's a link on YouTube videos in the FAQs. And then finally, on Saturday, the California Master Beekeepers Program is having a 2-hour class on installing package bees. That's not free, and we're not affiliated with the California Master Beekeepers, although that's where I'm trained for my Master Beekeeper certifications. Anyway, so that's where you get your information. We're planning on April 2nd for delivery of the packages. Do not arrive before 11. And the first thing you do when you get your package, you get it home, you put it in a cool place, and you pull out, you follow the videos on how to get your sugar syrup out, your queen out. You look at your queen first thing. She should be alive and running around. If she's not, do not proceed any further, and you'll call me in that case. If you don't call me and you install the package, well, I'm sorry, you're gonna have to buy your own queen. And finally, this is an example of how to install packages. These are all YouTube videos. Some of them are about how to do the shake-in method. The others are for the pheromone method, and you should, be teaching yourself on how to do this. This is not. We're not responsible for your bees once you get them. You need to learn. Teach yourself what you need to know in order to get the packages installed safely. Okay, and that's all I have.
All right, thank you, Bob. Hi, everybody. We have Dr. Ramesh Sagili tonight, who is an associate professor at Oregon State University. Dr. Sagili, are you able to turn on your camera and mic?
Yeah, I'm here.
Hi. We're excited to have you.
Yeah, thank you. Thanks for the invitation. Yeah, you want me to share my screen? I think maybe.
Yes, please. You should be able to share your screen.
Are you able to see the screen now?
There it goes. Yes.
Perfect. I will enlarge this as well. And probably now you see my full screen.
Yes.
Yes.
Perfect. Okay. Shall I start, or you have still some business going on, or?
No, please start your presentation.
Yeah. Thank you very much, and thanks for being here. I know it's late for you guys too, and I think this is my first time with your group. So I know few of your group members probably have seen me speak at the California State Beekeepers Association before, but I don't think I have spoken to your group directly. So yeah, I'm excited that I'm able to share some information both on bee nutrition and Varroa management. So you see, I have a nice English accent, so probably it'll keep you awake for the next 50 minutes or an hour. So how much time do I have? I mean, I am notorious. I can go for several hours and keep you here until midnight, but I want to respect your time. So can I go for an hour max, or?
Yes, yeah. So an hour, and then we usually leave about 15 minutes after that for questions.
Perfect. Okay. So yeah, I'll stop within an hour and then take questions. Yeah, I have too many slides, but yeah, I'll try to go fast on some which I don't think are that important. But I was ambitious. I wanted to give you more information on both topics here, bee nutrition and Varroa. Both are important, especially new beekeepers. I'll keep you in mind. I know some of you, I listened to some of your questions in the last 15 minutes, and looks like there are several of you who are brand new and you just want to start your bees this spring. So I'll be mindful that I don't go too much deep so that you are lost or something. Okay.
Just to that, the very first part of our meeting is for the new beginners who are there, but there are many people here who have years and years of experience. You can go as deep as you want to.
Okay.
There, some of us will find it interesting,
Yeah.
and the others will go, "Oh, those crazy beekeepers are saying things again."
Yeah. So I'll be careful about the veterans. Okay. So yeah, before I start, I always acknowledge the contributions of all these amazing current and past lab members that you see here. So I have been at Oregon State University, Corvallis about 12 years. And so our lab focuses mostly on honeybee health, which is a broad topic, including mites, nosema, anything you name it, including bacterial diseases. And then bee nutrition is the major area that we research. And there are only few labs. You'll be surprised when I go into this nutrition topic. I'll say that again. Honeybee nutrition is one of the understudied areas. Even though honeybee has been studied, more than 200 years in a more robust way, in a more modern world, but still we have a huge gap in knowledge on what bees really need. So I'll go into those details today as well. So let's start with the nutrition. You all probably are tired of listening to this before, this adage, you are what you eat. I know it's not just for us. I think it's relevant to our bees as well. So I always say nutrition is the first line of defense. We have so many stressors, and those new beekeepers, you will see not just Varroa. Varroa is your #1 problem, but there are so many other stressors, including nutrition, pesticide exposure. I'll talk about at least a couple of them today. So nutrition is the first line of defense. At least we have some control on this if we are able to feed them in time. And I think Robert was talking about feeding sugar. And so there are ways to feed your bees protein supplements if they need, depending on where you live. So I'll go over those details in the next slides at the end of the nutrition talk. So there have been a lot of studies that have already shown, so it's not something new. If you have better nutrition, then your immune system, that means your bee's immune system will be in a better shape, and then they will have a lot of detoxifying enzymes that can detoxify at least some of those toxins that they are able to get in, like pesticides or anything that you can think of. So this is a very basic or broad explanation of honeybee nutrition. So on the left, you have macronutrients. For the new beekeepers, this is what Robert was talking about, your sugar feeding, but basically once they are strong, they will collect their own nectar and honey, right? So they will collect nectar, process into honey. So that's their carbohydrate source. That's what I show you a honey frame here. And then protein, their only source of protein is pollen. So bees can't get pollen or protein from anywhere else. You have a lot of sources, right? You eat beans or meat and you get your protein. But unfortunately, bees are only reliant on pollen for their protein needs. So this is a pollen or bee bread that is packed in the cell. So here, this is, we know pretty much of this, right? I live in Oregon and we tell our beekeepers, I know you guys are in California, so your needs are different. Your bees probably don't have real proper winter like we have here, being in a more temperate latitude here. So we need at least 70 to 80 pounds of honey to survive the winter. So there might be some numbers, and again, you're in a good group there, so you probably get more regional information. I'll not tell you how much pounds or how many pounds you need in your area, but so that's what the overwintering process is. So we know all those things, how many pounds, and we know how much pollen bees collect and those things. But this is the area that we have a huge gap in knowledge, the micronutrients. So micronutrients itself tells you that they need only in small amounts, but they're really, really important. And micronutrients can be, again, vitamins. There are a lot of minerals and of course, lipids. And I'll talk more about sterols today. Sterols are a type of lipid, and they're really, really important for our bees. So just an overview of pollen. As I said, it's a protein source, and then you can see all pollens are not made equal. If you sit next to a hive, you'll see your bees bringing in, especially new beekeepers. You might be curious. I know new beekeepers are very passionate. They want to learn everything, and so they are going every day into the hive, probably sometimes. So you can sit next to the hive, and you'll see there are probably 5, 6 different types of pollen. Based on the color, you can tell, right? Depending on where you live. If you live in an area there is not much foraging resources, probably they're struggling and getting only 1 or 2 sources. But bees have evolved in nature for millions of years. They can really detect which pollen is good for them. So that's a reason, and each pollen is different, right? So different means not entirely different, but they may have some amino acids missing in one pollen, they may have in the other one. So that's a beautiful system bees have. They compile all these pollens from different sources, and then they pack into the cells in the comb, right? So they have access to different pollens so that they have a more balanced diet. And that's why having a pollen diversity is important as well. They can't just rely on one pollen for a long time. So you can see a range here. I've said 10 to 40% range. So some pollens could be 10%, and they will be in between all these ranges. And max we have seen is about 40% crude protein in pollen. So these are 3 things that you can think of depending on where you live, and it's same for commercial beekeepers. I know there is no one probably in your group who is a commercial beekeeper, but several things relate to both backyard beekeepers like you and commercial beekeepers, because we all have the same bees and they are facing same challenges. So lack of pollen abundance. So depending on where you live, when I say abundance, I'm talking about quantity, right? How much pollen? Is it 10 pounds is coming in a year? Is it 20 pounds? So that's what I mean by abundance. And then there is lack of pollen diversity that I mentioned. Like diversity means like how many different species of pollen they're bringing in. So some areas you may be, suppose if you're close to almonds in February, then probably your bees are only getting almonds. There is nothing else coming at the time except for a few wild mustard or some dandelions around. But we have done some studies where we see 95% of the pollen that comes into the hive. I mean, when beekeepers put in almonds, it's 95% almond pollen that bees are relying on. And then quality is also important, right? When I say quality, the numbers I showed you, 10 to 40% crude protein. So protein is one thing, but then there are sterols that I'll talk in a second. There are phytochemicals, there are minerals. Those things are important as well. So that makes the quality. So if you're in a crop like, I'm just giving an example from Oregon here, there is hybrid carrot seed across the mountains here, beyond the Cascades, where they grow about 5,000 acres, and it's a hybrid crop, and bees don't get good amounts of pollen when they are in carrot seed, hybrid carrot seed. And we have tested the pollen as well, and the quality is low. That means it's about 9% crude protein. But all your tree fruits and nuts, most of them are around 25% crude protein, which is a good number. Almond we have tested, and it's about 29%. So almond is a good source of protein. And that's why bees do really well when they are in almonds. When Oregon beekeepers or anyone in the temperate region, they go there, those hives are almost twice the size, the hives that they left behind in Oregon, because there is abundant pollen. Almonds, they may not produce that much nectar, but there is a huge amount of pollen that comes into those hives. So I'll quickly wrap this introduction here and then show you a study that we did. So again, as I said, lipids are also an important requirement for bees, which are one of the micronutrients. And among those, sterols are very, very critical. And I'll go over. So I'll start I know this is a Zoom presentation. I can't really ask you a question and get a response immediately. So think about your sterol. So we need sterols as well, right? Our sterol is cholesterol. So cholesterol, I know too much is bad, but we need cholesterol for our cell membrane development. There are so many functions that sterols have. So insects also need sterols. And there is a sterol called 24-methylene cholesterol. This is the most important one for bees. So that's what I say, it's a major sterol, and it comes from pollen. There is no other way they can get their sterols. And these sterols and this 24-methylene cholesterol may vary depending on what plant source they're depending on at a point of time to get pollen. So our liver makes our cholesterol, but for unfortunately insects, they can't get, their bodies can't make it. So they have to derive it from an external diet that would be their pollen. So that's why, just remember that sterols are important, and then they're also important because they are responsible for this molting hormones. If you remember your high school biology, you've probably heard of molting. So insects have to molt, right? They have to go to one stage. So suppose you have 1-day-old larva, that 1-day-old larva to become a 2-day-old larva, they have to switch, right? So there are some balance of hormones that happens. And that's what I have here in this slide. So here you see an egg, and then the larva, this is about less than 24 hours old larva. Then it's a little older one, and you see they keep growing, right? So from this stage to this stage, or from this to this, or this to a pupal stage, a pre-pupal or pupal stage, and an adult, you need these hormones. There are these two hormones, ecdysone and juvenile hormone. There is an interplay of these two hormones. So if ecdysone goes up and juvenile hormone goes down, then your insect switches to the next stage. So those are really critical hormones. And those hormones are really dependent on those sterols, the 24-methylene cholesterol that I mentioned, which is present in your pollen. So if you didn't understand anything that I said, just watch this animation. Hope this is working on your side. So that's the pollen, and those pink dots that you see, we are depicting them as sterols. So these bees come and eat the pollen, and the pollen again, the sterol gets embedded in their bodies, right? So you have this endogenous sterol. So the bee also has inside. She has internalized. She's feeding the larva now. So when she feeds the larva with the brood food, the larva gets the sterol too, right? So that's why she's able to grow, and then the bee comes out of the cell. But this is a scenario where suppose you're feeding just protein supplement, which probably doesn't have any sterols. So you don't have any pink dots, right? Because your bee didn't get any. And then she starts feeding, and now she's not able to transfer that sterol to the larva. So the larva will just desiccate, right? She will not grow anymore, and then she dies, and then the bees will either cannibalize her or remove her from the colony. So that's why these molting hormones are really critical. So what we did is, as I said, honeybee nutrition has not been very well studied, especially in terms of micronutrients. So we wanted to understand how much of this 24-methylene cholesterol is required, right? If you're feeding your bees, then how much do they need really? So we did a quick experiment in the lab where so this is a published work. So if you want to take a picture of this, or you can email me and I can send, it's an open access. You can just type in my last name, Sagili, and then just say 24-methylene cholesterol, probably you'll be able to find this paper. if you want to read, because I'm gonna go quick. If you need more details, then you should read this one. So here is the design. We have these bees in these cages. We provide them sugar solution. You probably have seen other speakers talk about research. That's how we do with cages. So they go into the incubator where we maintain temperatures like 33 Celsius and 55% humidity, like similar to what the hives are. So I'm talking in Celsius, so it would be probably close to your 92, 93 Fahrenheit. So we had these different diets. You see, we have 0.1, 0.25, 0.5, 0.75, and 1%. So these percentages are a percentage of total dry weight of the diet, right? So that's what we were feeding these. And because you may ask, why are you choosing these random numbers? Because we know insects, there is a range of sterols. So that's why we have different ranges here between low to high. And we wanted to see if bees prefer something within these ranges. So you may say, oh, so we can buy this thing. It's very expensive, so don't try it. There is not even a company that sells it. So we had to collaborate with a Canadian manufacturer. They custom synthesized this 24-methylene cholesterol for us. And then we use that. It's expensive. For 10 grams, they charged us $80,000. So I still joke, one of my postdocs who was working on this project, her hands would shake when she held a vial. If you drop, then $80,000 is on the ground in a second. So finally, we were able to finish this study, and here are some quick results that I want to show you. So we measured diet consumption, right? How much of the diet those bees in the cages were eating every week. And you can see week 1, these are the last bars here on the week 1. These are all high concentration. This is your 1% sterol, this is 0.75. So same letters means they're not significantly different from each other. So we didn't see a significant difference in the higher ones. So it looks like they all prefer, there is no big difference between 1% and 0.25 here. But there is significant difference between the controls and the lower concentration used. So you can see the similar trend in the other weeks as well. So we also measure this head protein. So head protein basically is we are chopping the heads and grinding the entire head to look at the protein levels. So there are 2 important glands, right? For the new beekeepers, you will learn the brood food is produced by these hypopharyngeal glands in the heads of the bees, and then they have mandibular glands, and the mandibles are the mouth parts, like the jaws of the honeybee. So those glands produce the brood food. So if we dissect, or if you take the entire head and grind, so you're getting the protein of both the mandibular glands and the hypopharyngeal glands. And that's what we mean by mean head protein. So you can see the high concentration sterols had higher head protein as well. So that means sterols are important for that production of hypopharyngeal gland secretions as well. So mean abdominal lipid, so you take the abdomen of the bee and then we look at the lipids in that as well. So you can see that we didn't find any big differences between these higher concentrations, but they're significantly different between your controls that didn't get sterols. So I apologize if you can't see very well this one, but there is an orange bar here on the top, orange line. So that's your 0.5 diet. So it looks like you don't really have to have 1% of the total diet. Doesn't have to be with sterol, but it could be 0.5, looks like is giving you better survival. So that was the major finding. So at least we were able to figure out what's the concentration that our bees need. So here is the take-home message from that study. So bees are able to detect, right? That's why they were preferentially eating the, so we had higher consumptions in the ones that were sterile. So this is a synthetic diet. Don't think there was pollen added to this. This is very, it was a challenge to make it attractive to bees because it's a synthetic diet. There is a mix of vitamins that we mix in there. There is Wesson salts, and then there is some sugar. That's the only attractive thing in the diet. There is no pollen in there. Because if we will put pollen, then you may ask, why didn't you add pollen? So pollen is a variable, right? That's what you have to do as a researcher. You have to remove as many variables as possible and just test the one thing that is different. So we just wanted to have a synthetic diet, which will not make any changes. And everything was same except for the 24-methylene cholesterol concentrations that we gave. Okay. I'll switch gear here and still nutrition. We are still on the nutrition topic. I'll talk about Varroa strategies and an oxalic acid study at the end, depending on how much time I have. But I'll finish first the nutrition part. So there is a lot of push, there is a lot of efforts, right? You have seen in California as well. If you're aware of almond production, there are farmers that are planting something for bees before almonds bloom, because bees arrive there 2 weeks before almonds bloom, and there is nothing else for bees to forage in late January or mid-January. So there is, it's not just California, right? In Oregon, there is some efforts going on in Midwest, and there was a memorandum, a presidential memorandum in 2014, you probably remember, where there was a directive to all federal agencies to encourage habitat. I don't think there was specific funding for it, but at least there was a directive to put more habitat for bees so that the bees could benefit from it. So there is a lot of push you might have seen in your neighborhoods with pollinator habitat, someone planting different types, and probably you are planting something for your bees as well. And in urban areas, that's very common. And even in, this is in Oregon, this is hybrid carrot that I was talking about. So we have tried to convince farmers to plant something like a hedgerow. It's hard, right? Because farmers, they say that we are high renting bees, we are already paying money. And there is other notion that if you plant too much here, then bees will be distracted from our crop and then we don't get good pollination. So there are things to consider as well. But if we explain the biology that if your hives are weaker and queen stops laying because there is no pollen coming in, then the pollination is impacted as well. And that's what we have been talking to farmers and explaining them some biology here, that the brood pheromone released by the larva stimulates bees to forage. So if your crop is not producing enough resources for them, then the bees are going to struggle and the queen will either stop laying eggs or slow down. And then the bees that are not stimulated will either be idle, sitting in the hive, nothing doing, and then their crop pollination will be impacted. So it's a win-win if they plant a small acreage. I'm not asking them to plant like, if you have 10 acres, I want 5 acres of supplemental forage, but a small 1/4 of an acre, or maybe even hedgerows like this will do the trick to some degree. So you may wondering why I'm giving you that introduction, because we are doing a study related to that. So here we got some funding from the United States Department of Ag last year. So it's a 3-year project, and we are looking for citizen scientists. So anyone from your group, after I'm done, you can email me and be a part of it if you think you can be able to help collecting some pollen for us. So I'll show you a video and give more details in the next few slides. So before that, again, so probably you heard that people ask you, or maybe you have asked someone, give me top 10, 5 or whatever plants I want to plant in the backyard so that the bees will benefit, right? And there are lists of, there are so many lists if you start Googling, and there is Xerces website, there is NRCS, Natural Resources Conservation Service, and there are some other groups that have their lists as well. But most of those lists are based on bee visitation, right? So it's basically, I don't think there is any empirical research done based on, suppose you say, oh, lavender is attractive. I see a bunch of honeybees coming, some bumblebees coming. Yeah, no one is debating lavender is not attractive. But if there is only lavender in your backyard, bees don't have a choice, right? And they will still come and collect nectar. But does that mean, and they will collect some pollen as well. Does that mean that lavender is the best source for your bees, right? Because we don't know the composition of the pollen of lavender or many of those species that our bees visit. So that's what I'm giving an example is, anything that is attractive doesn't always, don't get me wrong, sometimes it may be good too, but we want to answer that in a more scientific way. So does attractiveness always equate to best nutrition? So you give me French fries and salad, probably I'll start eating those French fries first, and maybe some of you probably too, because that's what they're attractive, right? Than your salad. And you know this is healthy, but still. So that's what I want to make. I know this is not a great example, but I'm just giving you some idea that, so right now we are all dependent on the attractiveness of those plants, but we don't know the composition of pollen. So that's what we are trying to do. So this is the NRCS website. They have a beautiful list of these plants. You can go into the fact sheet and find how can you grow those plants and what kind of irrigation they will need, what's the lifespan of the crop and all those, but unfortunately you don't have that information that you need for quality, right? So this is a Xerces Society website, which is here in Portland. They have a nice list of plants based on region where you live, and you can plant those. But this is what we are envisioning. We want to have this pollen nutritional composition. Like suppose you go into this, it says common camas, and you go here and we want to see, Okay, it has 10% crude protein. There is this sterol is present, 24-methylene cholesterol or cytosterol. And then we tell how many phytochemicals are there, what kind of mineral. So it's a huge task. And so we are envisioning to provide this information. And that's why we need pollen from all these different crops. That like we have about 100 in our list, but we are not stuck to this. If you think there is something you can collect in your area, we can look at that as well. So again, it's a volunteer thing. If you're interested, you can email me and you can sign up. I'll send you a video as well to watch and how to collect pollen from bees and also using hand. So we prefer hand collection, and we're trying to design a vacuum device to suck up pollen. It's been pretty challenging. No one has done this type of thing before. So we're trying to design a universal vacuum device which you can carry and suck up pollens from species where they don't produce enough pollen. Because we need about a gram. Gram looks like a small amount, but when you start collecting, probably it's thousands of flowers to get 1 gram. So that's our goal. Plant lists are not based on empirical research, and that's what we want to do, more of a scientific way to provide more information. So anyone that wants to grow these plants can have more scientific backing before they start doing this. So here is the video. I just want you to watch and hope it's working at your end. Any questions on that part? If you are interested, and so this is a bee part, right? You can collect using bees. So you have to watch the bee, and you have to have some significant, at least clumps or a significant acreage of a same plant species, right? If you have only 4 or 5 plants, probably you will not be able to catch enough bees. So we're looking about 50 bees loaded with those, their hind legs with pollen. So that would about give us a gram. And that can be used for all those different analysis that we want to do, including crude proteins, sterols, phytochemicals, omega-3 fatty acids, and all those different things. So this is my email. If you want to email me, I can provide you more details. I know it was a quick video. Probably new beekeepers are confused what exactly, but so either you can use bees for collecting the pollen, or else you can use a handheld vacuum. Once we come up with a prototype, then we will let you know. Or there are hand collection methods as well. They're painful. For some species, you can just bring in those flowers and then put in a flower vase with water and then put a white sheet underneath. And then by morning, you'll see a lot of, like sunflower is an easy one. You can bring in a couple of sunflowers and you'll get a lot of pollen. But not all pollens will, all anthers will burst open. In some species, it's hard. So there we want to use a handheld vacuum to vacuum up, and we are still in the process of designing. I don't have a vacuum right now, but we'll be able to provide you a design at least, or I don't think we can be able to ship that much because we don't have that much budget, but anything, if you can help with bees, that's great too. So, okay, if you don't have questions, then I'll move to the next. Okay, so you can see here, this is just for the new beekeepers. This is not that easy. Sometimes people say, our bees are starving, right? So you can see a well-fed hive. You should have all these larvae just floating in that beautiful food, right? The brood food. So you should have all this shiny food underneath. But if you see larvae that are barely nothing, so there is nothing, right? This larva has some, but these are like barely having anything. They're trying to move away from their actual positions. So larvae can move from one, I mean, it's hard to see, but if they don't have food, they will try to struggle and try to move around to see if they can find food inside. So the nurse bees are the ones that are feeding them and continuously providing this food. But you see these larvae, they are deprived of food. So this is not that easy always, but sometimes as a new beekeeper, if you find this, that means your hive doesn't have enough pollen. So that's why the nurse bees are not able to produce enough brood food, and they're not inside a pool of food. They're just struggling. This is something I just wanted to say, that the nutrition is important around the year, but I think the bees that I know you don't have a big winter like we have, or Minnesota or somewhere else. The bees are 5 months in a winter mode. So the bees that are produced later in the fall, we call them diutinous bees or winter bees. So they are the same bees, right? They don't look something like this here, but their physiology is different. They will have high fat bodies in them, and they have high vitellogenin, which is a glycolipoprotein, which is very important and for the longevity of the bees. So if you don't have enough pollen, then they will not have enough vitellogenin in the high fat body as well. So the only thing they do is so I'll give you an example how this works. So in summer, like probably in June, July, you have a lot of larvae, right? And you have nurse bees. They're eating the pollen and they're feeding them continuously. So they're not storing anything for themselves. Whereas when it gets to late fall, like August or September or October, whenever your queens are slowing down, so there is less number of larvae, right? Instead of 20,000 larvae, now you have probably, I'm just making up some numbers, you have only 200 larvae. But you still have nurse bees and still there is pollen in the hive. So they're eating it, and instead of feeding, there are less mouths to feed, right? So now they're storing everything in their bodies to survive the winter. And that's why in temperate weathers like ours, and the bees that are raised in October or late September, they are called winter bees and they survive until March. So we have done some studies marking bees and then watch them in March to see how many are surviving from August, September. I'll not go into those details, but just for the new beekeepers, just wanted to make sure you understand what those winter bees or diutinous bees are. So it's critical in the late season, your bees are not struggling with pollen. They need to have enough pollen in the hive so that they are raised in good nutrition, so that their immune system is robust and they will survive that winter period, whatever time it is, and be available in spring. So I know many of you probably use protein supplements, and commercial beekeepers, of course, they can't survive without using protein supplements, because they do crop pollination and they move around the country or they move around their own states as well into different crops, and some crops are good, some are bad in terms of nutrition. So they have to, and then sometimes in late fall or fall, they don't have much forage. So they do rely on protein supplements like this. And so if you are feeding, depending on where you live, if there is a need, you should. Protein has value. There is a lot of debate. Some people hate feeding protein. It depends on what it is, right? So I think the most common one is the brewer's yeast mixed with some things like sugar and some oil they use as well. So that's so this is one type. I'm not gonna give recommendations here as a researcher. I'm not supposed to give you any companies' names or anything because it would be a bias towards someone. So there are protein supplements depending on what your group may have used some before, and you can get recommendations from your local group. But protein supplement feeding has value. It's not waste of time and money. People have made this for the last 30, 40 years. So that some research has gone into those, and they are promising at least you can't survive your hive on protein supplement for several months. But at least it's a stopgap thing. Whenever there is a lack of pollen, there is a pollen dearth, these protein supplements can be really valuable. And then they don't store it. I know some people think they will store this, and they will never store. And we have done some work as well, and there is some research from other universities as well. So this protein supplements, your nurse bees will directly eat it. This you will not find this in your combs. They don't store it like your pollen is stored. So this is just a overview of what I said, some of those points that I wanted to make sure you know how important nutrition is. And so make sure your bees have access to adequate pollen throughout brood rearing season. Use protein supplements. The supplement I showed you before, some companies sell it with 5 to 10% pollen mixed in. I would prefer that rather than straight synthetic or something like soybean flour or brewer's yeast without pollen. May not be that valuable. It may still provide crude protein, but having a mix of about 5 to 10% pollen is really important because of those sterols that I mentioned. You don't need a ton of pollen, right? So in the next slide, I'll show you how you can trap some of your pollen, because when I talk about nutrition, people ask, can we trap our own pollen and use it in the next season or so, or in the same season as well, late in the fall. So I'll show you that too. So prefer always 5 to 10% pollen mixed supplements rather than just straight supplement. I think this I told you already. It's important when they're raising those winter bees, your bees have to have good nutrition, and Varroa management, of course. So here's a quick slide to show you. So these are front porched pollen traps. Probably some of you have already used them. There are different types. You have this bottom mount where it goes underneath the hive, but these go in the front. I don't think I have put a picture here, but these go in the front of the hive here. So these are easy to install. You can just either staple them or screw them or whatever, easy, or tape them as well. If you don't want to have any damage to your hive, you can put a nice tape as well, which will stay at least if it's not raining. And then what I want you to is select strong colonies. Just don't put traps on random hives because pollen trapping is an intense thing for bees. They will not have access to enough pollen for a few days, right? So you have to trap only when there is abundant pollen coming in. So you may ask, how do you know? You sit next to the hive, and it comes with experience, and see what time of the year is a good time when a lot of pollen forages. So you can sit and count. Maybe in a minute you are getting like 50 pollen forages. So that's the best thing you can do. Then choose those colonies and then install this trap. And you see this mesh here on the, I hope you're able to see my cursor. So that's the part where the bees will enter through those holes, right? So this is, it moves, so it can be flipped and attached to the top with a tape. So then there will be no trap, right? Bees can directly fly in. So you have to train your bees at least for a day. And then next day before they wake up, then you release this and it will be like this, right? They'll come through these holes and they'll think, oh, my weird beekeeper has put something around here. I need to go through these holes now. And they will come out and they will orient. So this is helping the bees to orient. Others they'll be confused that something is in their entrance now. And so that helps actually. And then this is a tray where you'll get the pollen. So this goes underneath here. So once you buy it, you'll understand how this looks like. And then you can trap for 2 days, I would say, no more than 2 days, because you don't want to deplete their pollen. Then your brood rearing will be impacted. So the colonies can get weakened. So you can again switch after maybe 10 days or 2 weeks. You can again put for a couple of days, depending on the strength of the hive, never on weak hives. And before you put this, I want you to close all the holes. You see this hive has an auger hole. We have auger holes here because we get a lot of moisture, we get a lot of rain. So you need to have those here. So if you have any holes or cracks, you need to seal them before you start trapping. Or else bees are pretty good. They will start using those entrances and you'll not be very successful in getting your pollen trapped. And then what else am I forgetting? Then once you collect in this tray, you can put them in Ziploc and put in your freezer at -20, and that might be good for a year. So pollen has been shown to be in good shape for a year. So you can use it for next year as well. And I'm looking at only like two tablespoons probably in a hive max with your protein supplement you're feeding, or you can even mush it if it's still in a good shape, and then just make a small patty and put in on the top bars, and they will eat that as well. So that's one way to provide your own pollen, so that you're not getting some contaminated pollen from someone else that you know, to avoid all those chalk brood and other diseases we are worried when you don't know the pollen source coming from a different hive from a different beekeeper. So those are some tips if you're interested in trapping your own pollen. And this is if you want to take a picture of this, or I can send a PDF of my PowerPoint slides as well. So this is a publication we published last year where you can read this to get a lot of tips on what I said is there also in that manuscript. You can read and figure out how to trap pollen because not many people do this. So, and we have learned this the hard way as well. Like especially commercial beekeepers, their hives, there is only probably an inch or two between two hives because they put 4 on each pallet, right? So there's a lot of confusion. If you put trap on one hive, then the bees from the other hive will all try, the one that is trapped will go into the next hive and you can have a different in strength. So for new beekeepers, bees are not always perfect. They will get confused and there will be a lot of drifting as well going on. So to avoid that weakening of the other hive, you need to put, so what we do is if we are trapping on a commercial colonies, then we put both traps for both hives so that you're confusing both, not just one hive. Okay, looks like I still have probably another 20 minutes or so. I'll talk about Varroa mites. Yeah, if you have any questions, you can ask. This is the time if you have any nutrition questions, but I'll leave some time at the end as well if you want to wait until then. If you have any pressing questions, you can unmute yourself and ask.
I have one question regarding the taste of the honey. If you feed with the protein supplement, it doesn't change the taste of the honey?
No, so protein is different than honey, right? So nothing contaminates your honey because this protein, these are nurse bees, which are 7 to 14 days old. They are eating that and they grow those glands and feed the larva. So your honey is a total different thing. Those are foragers that are collecting nectar from plants. So their job is very different. So they are never touching your protein supplement. So foragers will never eat pollen. It's the only nurse bees that are in the hive, which are very young, 7 to 14 days old. They are the ones that are eating the pollen or the protein supplement that you give them, right? So that's why there is no way that it will contaminate your honey. So don't worry about feeding. But yeah, if you're feeding something like your sugar solution and all, yeah, they will. The sugar syrup, they will store in the comb as well. So there might be something, if you think sugar is fine, then you should not be worried about it. But yeah, no, pollen or protein supplement doesn't contaminate your honey.
Thank you.
Yeah, Conrad has a question. Yeah.
If I see this starving brood, will pollen patties help? Will pollen patties and syrup help?
Yeah.
Or will nothing help?
Yeah, it depends how far they have gone without food. I don't think something magically will change in a day, but I would do the same thing. If I see my hives struggling and there is no fresh pollen available coming in, then protein supplements will help. It provides them some help with the crude protein. They may have a lot of other things missing, the micronutrients I talked about, but definitely it will provide them the crude protein which is required to produce that brood food. So yeah, it will help for sure. But again, depending on how sooner you catch it, if you are catching very late, they are already starving. So you may not see a significant improvement in a shorter period of time, but still I would recommend you feeding something like protein.
And I would assume the entire apiary is affected because it's the same location.
Yeah, depending on, yeah, that's a good question. So it depends on, that's what, when people look for mite monitoring as well, it depends on how many hives you want to check. But yeah, if you think, at least just don't make your decision just on one hive. If you have 10 hives, at least check 3 or 4 and then see that if they are showing similar symptoms, and then probably other hives you don't have to check. Maybe they are all going through the similar scenario.
Thank you.
Okay, I will switch to Varroa if you don't have questions, but yeah, feel free to ask me at the end too. Again, as I said,
one other
yeah.
one other question. Sorry.
Go ahead. Yeah.
So we often have frames that are packed with pollen over winter. The bees are basically making bee bread with, and sometimes people freeze those frames in order to prevent the wax moths from emerging. Is that pollen still good after it's been on the frame, out of the hive, in the freezer, then back out?
Yeah, so great question. So again, I haven't tested anything like that, but just based on my experience and logical explanation here, I would, if you have frozen them fresh, like taking from the hive, if they were not exposed to sunlight, or so the protein degrades only when you're subjecting them to high temperatures or sunlight, right? So if you have taken the frames immediately and frozen them, I would say if they may not be as fresh as when they were removed, but I would say they still have probably 80% of the quality still, because once frozen, as pollen. I think you may miss some of those microorganisms that were there. You know, when they are processing, there is some microbiome or bacteria. So some of that might be impacted because of freezing, but the pollen that is still there, I don't think the quality has degraded. So I think it's still worth feeding that pollen that is stored in the freezers. Okay, any other questions? If not, then I will switch. So Varroa mite, again, for the new beekeepers, I thought I'll just go some basics here. Those experienced beekeepers, I know you probably, this might be just a refresher for you, or probably very boring. So bear with me for the first few slides, and then I'll go into oxalic acid study that we did last year. So for new beekeepers, the mite is a major problem. So be mindful and don't get disheartened. I know I see a lot of beekeepers. We have a master beekeeper program here as well, and yeah, it's been probably 10 years now since we started the program, and I see new beekeepers, they lose hives. I mean, beekeeping is a challenge these days. You ask beekeepers that have kept bees before the mites were introduced accidentally into the United States about 1987. Before that, beekeepers had a relatively easy life keeping bees alive. Varroa has totally changed the face of beekeeping, and so yeah, even commercial beekeepers lose about 30–40% of their hives in a year. So don't get upset if you are a first-time beekeeper and you lose hives. I mean, that's how you learn sometimes. Beekeeping, you know, you can learn by reading books and also hands-on experience with your bee club or maybe some beekeeper program like your California Master Beekeeper program. But yeah, that's a story. Even at Oregon State University, we lose about 15–20% of our hives every year, despite a lot of management that we do take care of mites and nutrition. But that's the thing. We have a lot of stressors beyond our control these days. So don't be disheartened if you lose in the first year. You may be successful the next year for sure. So this Varroa is very important. So new beekeepers always monitor mites. That's a really important pest that you need to, that could break or make your beekeeping. So I'm not saying that's the only problem. As I said, nutrition is really important as well, but Varroa can really make a difference whether you are successful or not. So there are two phases we talk about Varroa mites. These are ectoparasites, right? They feed on the abdominal fat bodies of the bee, and they also suck up a little bit hemolymph as well. And then they have a reproductive phase when they get into the larva to reproduce. They can reproduce on the bee. So you'll see them on the bee. I mean, it's not easy to see unless you're an experienced beekeeper, you're looking at the right place on bees, but you have to do a alcohol wash or a sugar shake or whatever your mode of monitoring mites is. But you have to monitor. Monitoring is really critical. I'll talk about monitoring also again in the next slide somewhere. So basically what happens is these mites, let me show you a video here, animation. And so when I say phoretic phase, that means this is a mite, you see here. So if you make a fist and put in your body, that's the size. If you're a bee, and so that's a big parasite right on you. If I put a parasite of my fist, it's a huge one. So that's what these mites are. You see, this is a bee. You see, there is a mite. There is a mite here. There is a mite here. So that's called a phoretic phase on the adult bees. And when they're ready to breed, they go into the cells, into the larva. They can breed on the bee. So we'll see a animation to give a good sense on what these guys do. So that's a Varroa mite life cycle. So here is your hive. You have bees, you have mites. There is a mite on this female bee here, worker, and then she goes down into the cell where there is a larva here. So she hides underneath so that the bees can't find her. And then once it's capped, the wax cap goes on, then they come out and start feeding and laying eggs. So you see the mite is laying eggs. So she may lay, the first egg is usually a male, and then she may lay 5 to 7 eggs that are females. And then the male, so there is an inbreeding going on. The male mite will breed with or mate with the other siblings, the females that come out in the next few days. So that's basically how they do inside. And once the bee comes out chewing the cell, so this is the female mite that went in, the mother, and then now she's coming out with a baby or an offspring. So this offspring may take another week or so to fully mature and start infesting another cell. So that's basically your mite cycle. So if this mite finds a drone cell, because drones have 24 days, right? The life cycle of a drone is longer than your worker bee. So they have more opportunity to mate. So there might be 2 mites along with the mother. So there'll be total 3 coming out of the cell. So this is a question for you. What do you think? If suppose maybe in June you go into your hive, I'm just asking you a question to think, you don't have to answer. So what percentage of mites you think are always inside a cell or in the comb inside those brood cells? And how many are there? Suppose you have 30,000 bees and there are 10 frames of brood probably. I'm just making up some numbers. So what I'm asking is think about what, suppose there are 100 mites, just to make it simple. Out of 100 mites, how many are there on the bees and how many are hiding in those cells and breeding? So that's what I want you to think. And I put some numbers here, but I'll give you the answer based on our study in a second. So we did these are very laborious work. Okay, not many studies have done this, but we thought at least, and this may vary in your region. So I'm just showing you some Oregon data here. So we looked at this Varroa mite population dynamics. So the goal was to estimate both phoretic and reproductive mites. So what we did is we chose 6 colonies. You may say, oh, 6 is a small number, but when you see what kind of work goes on, you'll appreciate I can't do more than 6 here. We had to literally kill all these hives, all the bees, and then look mites on the bees, and then on each of those cells, of those thousands of cells of brood in each hive, right? So it's very laborious. So 6 is a good number for these type of studies. So we didn't treat the hives from March through September. So these hives never got any treatment. And then we monitored the mites throughout the year to see what numbers they are. But then eventually we went on September 14th. And then what we did is we chose a day when it's a little cooler in the morning so that all bees, at least 90% of them, are still in the hive. And then we do this. So you see, these are the hives here, right? So we shake all the bees into this funnel, like the package bee thing that Robert was showing earlier. So this is a little bigger one here. We shake all the bees and they get into this bucket with alcohol and they all die in this bucket, right? It's a destructive sampling. Yeah, of course, for the sake of science, we have to kill a lot of bees sometimes. So you get all those dead bees in there, and then we take the frames that have brood, we take them to the lab and put in a freezer. And then whenever the undergraduate students get time, they will look into those cells individually and see if there are any mites. So this is a video. Hope it's working on your end. It's a messy process, but
another brood frame. Can we slow down? No, we're good. Eggs.
so she's checking on brood, and then we shake all the bees, and those brood frames go back to the lab.
Brush wizard.
Yeah, the students appear to have fun here, but this is not fun. The next part I'm going to show you. So these are the buckets full of those dead bees. So we have a method how we can subsample each and then look for, because all bees we can't do one time, right? So we have a protocol on digging through each of those buckets and take maybe 500 bees at a time, and then we have a shaker. I'll not show you all those details, but we do this, and then that's not that bad. But this is the worst part. These students are just faking that they are looking happy here, but it's a painful exercise to have those magnifying glasses and look into each cell and remove. So you can see those. I'm showing you an example here. When they pulled, some of them had like large number of mites, like within the same. There were 3 or 4 almost mature mites too. So that's how we look for those mites. So this is another example from the same study where you can see the entire mite family here. That's a late stage, almost close to closing bee that has mite family there. So these are the numbers that you're looking for. So what we got was 56% in the brood, that means inside the cells, and 44% on the bees. So, and this may change depending on the time of the year. So don't think this is something, a magical number that you should, but at least for the new beekeepers, I wanted to make sure they understand So what you can glean from this is when you use something like oxalic acid or any treatment that is short-lived, not like APR, which you put and you wait for whatever 56 days or whatever the time period is. But in this case, if you're using oxalic vapor, right, it only stays for a day or maybe that few hours that you used it. So then every day there are, if your queen is laying 2,000 eggs in a day, so there are no 2,000 new bees coming out every day, right, in summer or spring. And maybe if you think half of them had mites, so you still have about that half number of mites coming, right? If you have 1,000 cells and 1,000 bees coming, the 500 bees coming out with mites means there are 500 mites there. So you can do something like that. And so that's the reason I want to show you this study is sometimes beekeepers, new beekeepers may think, oh, I used oxalic today, so I'm mite free. You are not, because that day you are mite, at least you have reduced the numbers on that day, but there will be new mites coming every day, right? So you have to see that when you use products like oxalic, you have to use them frequently, at least a week apart or 10 days apart, thrice or whatever your regimen is. So that's what I whole study this. Again, this helps us do some modeling as well to see how many mites are there, and based on that you can choose some treatments as well. So there is some value to these type of studies. So this is the take home message. 50% mite at least are always there in the capped cells if your queen is laying vigorously. So these are some products. I know your group probably has already gone through many of these, or you learn about miticides that are available for you to use. I wish there was one silver bullet, but you don't. So you have to rotate between these chemicals and see. Some of them are temperature sensitive, so you can't use when it's too hot, depending on where you live. So there are a lot of limitations, but these are some products that work. So you can choose one of, or two of, or three of these as well. So I'll quickly go into the oxalic. I know some of you probably use it or are interested in using as a vapor. So we started looking at oxalic acid because there are a lot of questions about efficacy. Is it toxic to larvae? So we wanted to address some of those questions. And now oxalic acid, the new label, at least if you buy something from Man Lake or wherever you buy your product, Data or wherever, the new label should say that you can use when honey supers are on, right? That's what the new label should say. Oxalic acid can be used when honey supers are on the hive. So earlier there was a restriction that you can't use. Now it's available to use. So that's a label people have used as dribble. We have used at OSU for winter time. We use when the brood is all, I mean, a very small amount of brood or no brood, and there is a window where bees are all clustered. At least that's what happens in Oregon. So we use a dribble method. So we dribble oxalic acid on the bees, and that's very effective. But that time it's too late. If the brood is there, the brood is already damaged if you had too many mites and you are not able to control your mites. But any leftover mites, you can remove them by oxalic acid dribble, and you may start with less number of mites next season. So these were the questions we always wanted to know. 1 gram. So right now the recommended dose is 1 gram per brood chamber. That means if you have 2 boxes, you use 2 grams to vaporize it, right? That's the label dose. But there were a lot of complaints that it's not giving good control. So we wanted to see different doses and see if it's working or not. So, and then there is a concern like how many times you want to use oxalic, right? I mean, it could be toxic to bees and the brood if you're using too many times. So we wanted to see what we can do. So we used 3 times in our study, like 1 week apart. And so we wanted also both brood damage, any to bees, and then this is another interesting one. We usually get our queens impacted by oxalic acid vapor. So we wanted to look at that as well. So the first study, I'll quickly go. I know I'm running out of time here. Maybe I have another 5 minutes max. So we wanted to look at mortality of eggs, right, and larvae, and this vaporization. So this is how we use. So I would recommend always use. Your health is more important than your bees. Oxalic vapors are really toxic. You don't want to breathe in. So use this respirator if you use. Never without a respirator. So that's a 2 gram dose or 1 gram probably. This that Carolyn here, one of my lab members, is demonstrating. So we use a spacer here. You see that spacer here between the box and the bottom board here. So this allows you not to burn frames, because some beekeepers I have seen, they have put this extremely hot wand inside, and the bottom of the frame touches it, and you have created a hazard in the hive with fire. So please use something like a spacer. It helps the fumes to go uniform as well, right? If you're touching a frame, it not only creates an hazard, but also the fumes may be not going up. Maybe you max the wax melts on it, and then it may not be effective. You want those vapors to uniformly spread in the hive. So always use a spacer so that there is space and your wand doesn't touch your, depends on what type of device you're using. So we have a master beekeeper program, as I said, so we provide some of those master level students opportunities to interact with us and help with the research. So you see here, these 4 gentlemen here trying to take data on these acetate sheets. So we put the pins here, and then we match those cells, and then there are dots. If you can see in the next slide here, see those red dots. So we are marking those larvae, like 50 of them, which are 4-day-old, 50 that are 2-day-old. And then we come back every day and put that sheet back on, and then see if that larva is gone because of oxalic acid vapors. Did the bees remove it because it's dead? So that's how we measure very precisely the mortality of those larvae and eggs. So it is a quick overview. So we also used formic just to see. Formic is relatively toxic, you all know probably. If you have used formic acid, the Formic Pro or the Mite Quick Strips in the past, formic is really toxic, but it does a real good job of killing mites, but you may lose some brood as well, depending on what time of the year you're using and how much ventilation you have in the hive. So I'm saying formic is a good product, but use it carefully. So here you can see in 24 hours, this is the formic acid, about 30% of the eggs are gone. Whereas there is no big difference between your oxalic and the control. This is your control, that means no treatment. This is your oxalic. So you can see a similar trend here, but here you see after 1 week, formic, there is about 85% gone, the eggs. And here, these are not significantly different. Then we looked at the young larva, right, 2-day-old, same thing. You see formic is still killing about 20% plus of the larva in 24 hours. And then after 48 hours, if you look at the data, about 60% of the larva, but then there is not much difference between oxalic and controls. This is the older larva. So older larva looks like they're more resilient. So you can see, so you see the scale is smaller here, right? You're losing only 5% or less in formic. And even after a week, you're losing only about 15%, 24 something for formic. And there is not much difference between control and oxalic. So take home message, oxalic acid vapors at the recommended dose of 1 gram per box look safe. We haven't seen much of a problem. They were relatively same as the control groups. But then we don't think 1 gram is working. So we did another study. So looks like I'm almost on time, but yeah, if you can bear with 5 more minutes, I'll be done. So 2021, we did another study. Here we wanted to test 3 different doses. The 1 gram is the label dose, right? Then 2 grams and 4 grams per box, right? So this would be 8 grams for 2 boxes. That's very high dose. And 3 applications, 1 week apart. And then we measure all these mite levels with alcohol wash, sticky boards. So we look at the colony evaluations like number of bees, brood. We look at some physiological parameters as well, looking at the oxidative stress on the bees, any damage to their legs and mouth parts, like appendages. And then we also collected queens at the end of the study to see if they were in good shape in terms of their sperm viability, and which is still a pending one. We haven't finished that study yet. So this is how we do. You see, because we use 2 batteries, because now you're burning too much of a dose, right? I can't put 1 gram or 2 grams at a time. That's not a good design. So we put 2 wands at the same time and burn as much as we can, so that we don't have to do twice or thrice. And then we put this wet towels outside so that the fumes stay inside at least for 10 minutes. If you don't put these, then the vapors will come out and it may not be very effective. So I would recommend, if you're using oxalic with a battery operated device, put the wet towels at least for 10 minutes so that the fumes stay inside for that 10-minute time. Then you may have a better control. This is a video, but maybe I'll skip it. Let me see. See some fumes coming out there. So that's how we do. So there are two devices here. There is one that has gone in here and one here. So we are burning about 4 grams right now here. 2 grams in this one and 2 grams in this one. Okay, I'll be real quick on these. I apologize. I know I have too much information to share, but we don't have that much time here. So, so you can see here, or maybe I'll just go to the final slide. And this is important, so let me not skip this one. So we were monitoring those mites on a sticky board as well, right? We do alcohol wash. That's the best method for new beekeepers. If you really want to know your mite numbers, alcohol wash is the best method. Sticky board is relatively easy. You just put a sticky board on the bottom board and then monitor mites every 24 hours or 48 hours. That gives you some sense, but it's not a very accurate way to figure out. So here, this is a sticky board data that I'm showing. So here you can see that's your 4 gram dose, right? The highest one. So you see there are still about 700 mites are being dropped in 24 hours. And then by day 2, I mean, end of day 2, you are looking at about 180 something still dropping. And then day 3 also you have some drop close to about 100 mites. But then after that, it just plateaus. So you don't see much of. So at least this tells that oxalic acid doesn't just stop in a day. It works until at least day 2. But most of the drop is happening by day 1 after 24 hours or within few hours after you put the oxalic acid in. And then you can see that's the top one is your 4 gram. The orange one is your 2 gram. That means 2 gram per box. And then you have this 1 gram. See how what kind of difference here, right? 1 gram is not giving you that much good control here. So then you have, this is another second treatment. That was first treatment, right? Because I said we use 3 times. This is the second one, similar data after 1 day. You're still dropping that many mites, 1,000 plus. These are the same, again, this is the average of all those hives that we had, right? So 2 grams, you're seeing about 800 mites still coming down. This one is your 1 gram, the recommended dose in the label, not so much when compared to these. This is your control. So oxalic acid vapors appear to be relatively safe to bees and brood if you use that 1 gram dose, right? If you're going up, then there is some damage for sure. So I didn't show you all that, but I can answer the questions. The current label dose 1 gram appears to be inadequate. So I don't think 1 gram. So that's what when we publish, and there is work published from University of Florida, if all those publications get available, I think there might be a change in the label dose probably in the next year or 2. Depending on when EPA wants to change the label. So 1 gram, of course, is not working as intended. So oxalic is, again, I'm not saying it's not working, but it's a very important tool. So that's what I'm saying. What I mean by IVM is integrated Varroa management. So you can use oxalic whenever you have a window to use. I recommend using oxalic acid, but maybe at a higher dose, but then again, that would be a violation of the label, right? So you are in a sticky position right now, but yeah, I think that's the story of oxalic. So I have some quick strategies here. I can't emphasize the importance of monitoring. For new beekeepers, please monitor your mite levels from spring on until late fall, at least every month, so that you have good handle on your mite levels, because mites can make or break your beekeeping. So instead of a calendar-based time, I know in Oregon, I hear some beekeepers will say, "Oh, on Memorial Day I'll do this, and Labor Day we'll do this." Don't stick to calendar days; those days are gone. You have to monitor every month with alcohol wash and see what your levels are. If your brood reading starts early in the season, like this year, we didn't get much rain in Oregon in February, and we see a lot of brood already. So as I said before, as soon as the brood is available, the mites are having a feast, right? They know now they can breed inside those cells and increase their numbers. So if the brood season is early, then you have to monitor early and keep those mites under control. So use combination of products, don't stick to one. Like APR works very well in the spring. That's what we have seen when the mite levels are low, but when the mite levels are too high in late summer or fall, then APR doesn't do as great job as it does in spring. So here I'm just giving, don't stick to this example, but I'm just showing something that has worked well for some commercial beekeepers here in Oregon. So they use Amitraz during spring, and then formic acid during summer and fall, whenever the temperatures are reasonable. If it gets to 100, no one is going to use it. But they're using a single pad. So there are two ways to apply Formic Acid Pro, right? There are 2 pads for 14 days or 1 pad every 10 days for twice, right? So that's how, so 2 pads is relatively toxic to brood. So if you don't want to kill too much brood at a time, especially when they're raising those winter bees, then probably good strategy is to 1 pad at a time. So rotate after 10 days. And that's has been working well. And then some beekeepers are using oxalic vapors whenever feasible in spring and summer, depending on their convenience, when they have extracted honey or not. But right now, oxalic, based on the label change, you can use it even when the honey supers are on. So this is the most important part. And many beekeepers don't monitor their mites after the treatment. So if you do a treatment with APR or oxalic or Formic or your AP guard, which is the thymol, I want you to monitor after a month after the treatment to see if the mite product has done any good or not. Because if you blindly believe things are okay, then the damage is already done, right? After a month, you if you're not doing anything for another couple of months, the mite numbers will just explode. So be careful and be monitoring the mite levels after the treatment as well, not just before. The before treatment tells you that you should start doing something, and then after you use a treatment, also monitor so that you have a good sense on if the product is working or not. The last one here is if you get, I know there are a lot of people selling queens. I don't know where your source is, where your guild gets their queens, but see if they are selling something, they claim that it has mite resistance. There is not real resistance at this time, but there may be some stocks that are a little better in tolerating mites than others. That's another strategy, but I can't recommend anything. I haven't seen anything that is really tolerant or resistant to mites at this time. Maybe in the future, there is some breeding programs going on to look at them. Okay, I think that was my last slide. I'll stop here with my dog pictures here, and I'll take any questions you may have.
Thank you so much.
Hey,
Anybody?
Ramesh.
Go ahead.
Ramesh, for Formic Pro, you showed what the brood like loss was during treatment and after treatment with Formic Pro. Do you have similar data for oxalic acid?
Yeah, that's what I skipped, right?
Oh, any chance
Yeah.
you could either share the slides or go back?
Yeah, let me see if I can.
Thank you so much.
Yeah. Yeah, I apologize. You know, I want to respect your time. That's why I just skipped some of the slides, but I do have the brood.
We're happy to get good information.
Okay, so yeah, if you are ready, then I can still go for another 5. I am not in a time frame. I committed for this talk, so I'm not in a rush. I just want to respect your time. And let's see, that's a mite. So you can see this is the mite data that I skipped. So this is your control. You have 1 gram. So this is a percentage change. So you have to understand what I have on the y-axis here. So that means the mite numbers have changed. So that means it has increased to 8%, right? That's what it means. So if you use 2 grams, the numbers have gone up to 6%. And with 4 grams, you see there's a very good control. The mite numbers have not jumped that much in those times. Let me show you the brood. So that's the number of frames of bees. And yeah, this is the brood. So here in this case, so I showed you the first study, right, where we were meticulously looking at number of larvae. Like we had 50 age group up to 2-day-old, 50 larvae from 4-day-old. But in this one, we were looking at frames. Like we measured the area of brood area in those, right? So that's what it says here, number of frames of brood, right? So you can see 1 gram here. So this is a lot. How many? So this is all in minus, right? What that means is that much of less brood is there, right? When compared to that. So in control, you're losing. So it's not a real good comparison because controls will mess up here, right? Because we didn't do anything for mites here. So that might impact their brood as well, because the brood is impacted because of mites, because we didn't do any treatments on controls. So don't pay attention to the controls, but you can compare these for sure. So you can see there's 1 gram here. You're losing about a 0.8 or 0.9 frames of brood for 1 gram. For 2 grams, you're losing about 1.1 frame. For 4 grams, you're losing about 1.6 or 1.7. So 4 grams looks like is impacting a little bit more, right? 2 grams looks like. So what we are trying to do this year is we are trying somewhere between here. So this summer we have a plan to do a 3 gram, which I'm imagining that it might be a best one, which is not killing that much brood, but still giving a good control. So does that make sense?
Yes. I assume that these colonies were in double deep for the
Yeah.
brood chambers.
Yeah, they are all double deep. Yeah.
Okay, so when you say 4 grams, okay, yeah, all of that makes sense.
So yeah,
Thank you so much.
so this for, I mean, yeah, I know this is confusing. When I say 4 grams means this colony received 8 grams total.
Exactly, and it lost 1.6 out of 20 frames.
Yeah. So if you're if you do that math, yeah. So this is 8 grams going into those 2 boxes together. Yeah, I can share this PDF if you're interested. I can email, and then you can look at your convenience here.
So on balance between brood loss and efficacy, would you recommend 2 grams?
Yeah, that's what we are trying to look at, that soft, the balanced place where, that's why if I do 3 grams this year, if you ask me this question next year at this time, I will have a good answer for you.
Okay.
So we want, I think there is something in between here where we think is a good goal. Like 4 gram looks like it's killing a little more than we were expecting the brood, and 2 gram is not statistically. If I don't think these two are different here. So I think if you go 3 grams, maybe you'll end up somewhere here, and maybe that's a goal where you get a decent control and also not kill that much brood like farming does. So that's the goal. So we are not using 4 grams anymore. I am discouraged because of this brood impact here. But I think 3 grams might be the one that can do the trick. Or else, so safely at this point, based on this data, I would say 2 gram probably is working at this point. But yeah, it's not giving you as much mite control as 4 gram, but maybe 3 grams. But again, I'm not the one recommending it. It's the label has to change. So hopefully our data, and then University of Florida, Dr. Jamie Ellis, they have published some work last year. Actually, Cameron Jack, he was my student. He did a master's with me and went on to do a PhD, and now he's an assistant professor. So he did the study, but they used only 1 box. Florida is weird. They only go with 1 box. I think that's common there, I guess.
That's not the only reason that Florida is weird.
Okay. So
this is all, I mean, the science is there. It's great. In California, we have an issue that the government says that we can't use oxalic vapor.
How come?
At least that's the law as of last year. I haven't checked lately, but
No,
It's still correct.
Interesting. So they don't go with the EPA label that is available? Do they? I mean, California Department of Ag has different set of restrictions?
Yeah.
Correct. Yeah, they take the federal ones, and it's even more, like most things in California, take the federal law and crack it down even more. That's what California gets.
Interesting. I didn't know that. Yeah, in Oregon, yeah, it's allowed. I mean, our beekeepers do use. So, yeah, that's the thing. Oxalic is a tricky one, but hope the label changes and you can legally use it at some point. I mean, is someone trying to convince California Department of Ag to?
Yes.
Okay.
Many groups are, and it being illegal isn't stopping a lot of backyard beekeepers from doing vaporization anyway, of course.
Yeah, I know.
It's really only stopping the commercial beekeepers who mostly probably won't use oxalic anyways.
Yeah, it's not that, it's not convenient for them, but some of them are interested. I have seen some Oregon commercials that have started using at least once in a year. But yeah, it's not that easy. The commercial for hobby beekeepers, yeah, it's relatively simple. If they follow all the guidelines, and yeah, still the vapors are toxic, and I really want whoever uses it to be careful. Use a respirator for sure, or if there is a way you don't breathe those fumes in, they could be pretty toxic.
Then
a quick follow-up question.
Sorry, go ahead.
I believe you mentioned that you treated 3 times in these tests with oxalic acid. How many days apart were those treatments? Was that the normal 5 days or something different?
1 week apart.
1 week apart. Okay.
Yeah, we were struggling, like, I mean, because this is a random design, right? There is no guidelines on how, I mean, no one is telling you how many times you have to apply. And so we did some survey with beekeepers, how often they were using. So some of them said 5 days, 7 days. So we thought 7 days is somewhere ballpark, which is more reasonable. And then if you look at the brood cycle also, that's another criteria, right? People think 21 days is a brood cycle. So if you do 7 times 3, that's pretty close to that brood cycle as well. So
looking at your data, it looks like you should do it every like 3 or 4 days, every 4th day, to ensure that you're hitting all the uncapped brood while it's still effective. Because it looks like that 4th and 5th day, 6th and 7th as well, were nowhere near as effective as the first 3 days.
I'm sorry, but you have to remember that you're not treating from the 21 days. You're only treating what's under the cappings, and that's a 13.5 day for a drone. So that makes sense to this every 7 days. Although you're going to have workers or drones emerge in that time frame, and the sexually immature mites are going to be mature in somewhere between 5 and 13 days. So if you treat every 5 days, you're going to kill those sexually immature ones as well before they can go back into a new cell and start reproducing for the first time.
Yeah, that's a good point. So I think all the data that, we're still in very initial phases. Once we have all this data, I think that might be useful to even figure out a perfect regimen, like as you said, 5 days, or as Levi was saying, 4 days. So I think there is a way to figure out those, and maybe you can go 4 times probably rather than 3 times and see what is effective. But those are some things we can play once we figure out the exact dose that is really impactful.
All right, Lynn Rousseau asked, are there any effects from oxalic acid on queens? I think you said that you're running a study but don't have results.
Yeah, we did collect queens, but so we don't do sperm analysis. I have a colleague, probably you all know, Dave Tarpey. He's in North Carolina State. So they run a queen clinic there. So we usually send all samples there. So we have sent the samples, but we haven't gotten the results back. So we are looking at mostly in queens. I only am interested in sperm viability, right? So does oxalic acid impact their sperm quality, and if that impacts their brood lay, the egg laying and all that. So that's what we want to look at. So once that data comes back, maybe next time if you have time, email me and I can tell you the impacts on queens.
Sounds good.
I have a question.
Go ahead, Naomi.
Did you ever do any comparison between bees that are better fed with the Varroa, whether it really affects the resistance? Because you were talking in the beginning about the immune system. So did you do any comparison? You understand my question?
Yeah, that's a good question. Yeah. So I have not directly done, but there are a couple of studies that I'm aware of. They have, it's very tricky because you can't control some things, right? Just the diet, and then if it's an open experiment in the field, there are so many other variables too. That's what I know. We have a very fascinating insect, but it creates a lot of complexity for us to control variables in the field. What I do in the lab probably is not very realistic if it doesn't replicate in the field, right? That's why you see with a lot of products like RNAi or this metarhizium that they're trying to control mites, and it doesn't work in the field because of a lot of other challenges. So coming back to your question, at least those couple of studies I'm aware of, they have shown some promise. If your bees have at least better nutrition, they are able to tolerate mite levels to a certain degree. I'm not saying they'll be okay, but at least there is a difference in some physiology that they have looked at.
Thank you. So is it getting worse and worse by the years?
Yeah, so I think, nutrition, if you can control, because Varroa is just not the only problem, right? They are not only feeding on the abdominal fat bodies and hemolymph, but the major thing, the damage they're doing is transmitting the viruses, the deformed wing virus. That's the major challenge. We see that all the time in the hive, and mites are the ones that carry that virus and transmit to other bees. And so deformed wing virus is there in other countries too. And even in the US initially, before the mites came, we still had the virus. But then the virus got another mode of transmission. Now the mite So before that, they had this oral transmission. So if I'm a bee, I have the virus, and then if I do this prophylaxis exchange of food, then I get the virus. So that's one mechanism. But now the mite is the carrier. She goes and like you going with a needle and poking everyone with the virus. So that's what is happening. The mites are another mode of transmission, very one, and that has made some viruses more virulent and more problematic in the last few years.
Thank you.
All right, I think we can do one more question and then we'll let Dr. Sagili go. Does anybody else have a question?
Thanks a lot for speaking with us.
Thank you so much.
Yeah, no problem. Yeah, it's a pleasure. Looks like you all have great questions and a lot of enthusiasm. So good luck with your bee season, and hope to see you guys sometime again next year.
Thank you.
Thank you.
If anybody would like to hang on, we will raffle off two things today. So a quick raffle for anybody